Hello dear aspirants, welcome to GDC platform and I welcome you for my Pharmacognosy sessions. Yes, my dear students, now we are discussing, up to now we have seen different types of alkaloids. We have covered the introduction of alkaloids and we have seen various types of alkaloids, pyridine, bipyridine alkaloids, and we have covered some other alkaloids. Okay, yes, now in this session we are going to cover the rest of the alkaloids, that you can see right over on the screen. Especially in this session, we are going to start with a new and another important alkaloid, that is quinoline alkaloids. That is quinoline alkaloids. So I'll give you one hint. Okay, yes, queen. So what is the drug you have remembered? Meanwhile I'm speaking, meanwhile I'm pronouncing queen, cinchonidine, yes. So the drugs which are possessing the quinine nucleus, quinoline nucleus, comes under the quinoline alkaloids, which means they are possessing the quinoline nucleus. We'll see what are the drugs are there in the quinoline alkaloids, and also we'll cover some of the important points with respect to our GDC and with respect to our GPAT examination. Yes, before that, we'll see their chemical structure and occurrence of these quinoline alkaloids. Yes, because these are containing the quinoline ring. The quinoline alkaloids are class of natural compounds characterized by their chemical structure containing a quinoline ring. We have seen that the tabular form which we have that all the structurals, okay, all the structures and all these things. Okay, so coming to the chemical structure, yes, undoubtedly it's a bicyclic structure composed of one benzene ring is fused to pyridine ring. One benzene ring is fused to pyridine ring, forming quinoline ring. Forming a quinoline ring. Yes, but in this quinoline ring contain nitrogen atom which distinguishes the quinoline alkaloid from other alkaloids. Okay, yes, in this quinoline structure, if you have seen that the previous or starting introduction class, then that I have shown you that the alkaloid, the quinoline alkaloids having the bicyclic quinol ring. In that itself it is having the nitrogen atom. That's what we are discussing. Okay, so because that makes a difference between the other type of alkaloids. We have bipyridine, pyridine, indole and all the alkaloids, but what makes a difference? A bicyclic nitrogen containing ring, that is a quinoline ring. Yes, my dear students, mostly that coming to that occurrence or distribution of these quinoline alkaloids will be seen in a special plants, the special families, or the chemotaxonic feature of these quinoline alkaloids is 𝘂𝙬𝙎𝘾𝙬𝙎. 𝘂𝙬𝙎𝙬𝙎. So only in this species we can see the synthesis of the quinoline alkaloids. 𝘂𝙬𝙎, we have seen that cinchone and all these things, they are 𝘂𝙬𝙎 family containing quinoline alkaloid containing drugs. Okay, so some of the examples, like that's what we said, quinine. It's an quinoline alkaloid which is obtained from the bark of cinchona trees and which is used to treat malaria. We know that quinine, malaria drug, anti-malaria drug, and also 𝘾𝙀𝙉𝙀, that is the Chinese tree of 𝘾𝙀𝙉𝙀 acuminata, which is an anti-cancer property. So these are the main two families that we can get the quinoline alkaloids. Yes, coming to that, why I have introduced or why I have included these families or occurrence in this particular topic. Why? Because sometimes in the GPAT examinations they may also ask about the what are the distribution, what the occurrence, like in which families, in which plant family the quinoline alkaloids are there, the quinoline alkaloids are rich in, or which plant species accounts or accountability for the most of the quinoline alkaloids. Yes, undoubtedly the 𝘂𝙬𝙎 and 𝐹𝐴𝐶. Clear, my dear students? Yes, we'll see the biological effects. Yes, when that every drug, every alkaloid is possessing a specific properties, specific properties or specific biological effects. In the last session, just before this previous session, we have seen that almost all the Solanaceae family containing drugs are possessing, or the atropine and all these things are possessing almost all species having only one property, that is anticholinergic property. As like how these quinoline alkaloids also having some properties. Let us see. The quinoline alkaloids exhibits the diverse pharmacological activity including anti-malarial, anti-tumor, antiviral, antibacterial properties. These are their activity. These are pharmacological activities. And also, but coming to that, the quinine is the most important alkaloid which counts for almost all the pharmacological actions. Okay, so it affects you again. It's a plasmodium parasites that cause malaria. Yes, because of this particular quinine, the quinoline alkaloids came into limelight. The QUINOLINE that because has a very potent drug against the Plasmodium vivax or Plasmodium falciparum. Okay, so like how it interferes with interfering the parasite ability to digest the hemoglobin. Yes, it prevents, it interferes with the parasite's ability to digest the hemoglobin when it's the life cycle carries out in our body. Clear, my dear students? And the camptothecin, that's about the quinine, that's nothing but cinchona. But whereas the camptothecin, it's a derivative has been extensively used for their anti-cancer property. So that you should understand that the quinoline alkaloids are mainly for having two properties. One is anti-malarial and second one is anti-cancer properties. Okay, so it inhibits the enzyme, the topoisomerase leading to topoisomerase 1 leading to the DNA damage and apoptosis in cancer cells. So that means it topoisomerase enzyme, the camptothecin will inhibit the topoisomerase 1 enzyme, which can cause a severe effects or severe damage to the DNA. So, but thereby it prevents, it blocks the synthesis of topoisomerase. Thereby, there is no effect on the DNA and their apoptosis. That's what about the biological effects that is producing on the body. Okay, and coming to the medicinal uses of this quinine. The medicinal uses, yes, undoubtedly we knew that this is one and only source, one and only the medicine that we till now we are using, that is a quinine, the most potent and prominent medicine for the malaria. Yes, so that's the malaria, and although its use has been declined due to development of other antimalarial drugs. Yes, later on due to evolvement of after that due to the several researches, so this the use of the quinine or cinchona was gradually reduced, but it is still employed in certain cases which are very much the treating the drug resistant strains of malaria. Some species or some strains are very resistant to specific medicine. In such case also, such type of resistant strains also can be cured by the malaria. And whereas coming to the camptothecin will be having its analogs, that is irinotecan, irinotecan and topotecan. They are mostly can. You can remember that can means cancer. Okay, so that you use in a cancer medicine. Okay, cancer, topotecan cancer, irinotecan cancer medicine used in chemotherapy, particularly colorectal, CCC, canotecan, irinotecan, topotecan used in cancer medicine, especially to treat the C, colorectal, or C, colorectal, ovarian and lung cancer. And also sometimes it may helps to recover from the hepatotoxicity and when it's used in a cancer therapy. Yes. And coming to that, what is this chemical synthesis? Chemical synthesis, it is synthesized through a complex molecules like synthesized chemically, especially these molecules or this quinine are synthesized, quinoline alkaloids synthesized chemically. All these that we have discussed up to the quinine, quinoline and all other things are generally we used to synthesize them in a chemical manner, especially from complex complex product like quinine, camptothecin in a multiple steps. Okay. Yes, and many quinoline alkaloids are obtained by extraction. Yes, nothing but for how do we extract, how do we synthesize, which means initially we used to say extract from natural sources and other semi-synthetic sources by using a simpler precursors molecules. That's about the biological effects, medicinal uses and chemical synthesis of the quinoline alkaloids. Okay, yes, we'll see individual drugs from this topic so that you can understand, and you should remember the synonym from the synonym to the uses. First and foremost, we knew that cinchona. Cinchona is a bark. It's a very outermost dead bark which is used to treat the malaria, that we know, cinchona. But what will be the chemical constants? Quinine. Okay. So the coming to that, it comes under the quinoline alkaloids, and mainly, especially from cinchona, you should definitely remember the synonym, because in the synonym itself they have asked so many times. So Peruvian bark, 𝙬𝙎 bark. So they'll give you the synonym name and they'll ask you to pick the right answer for this one. Okay, so that 𝙬𝙎 bark and Peruvian bark. I tell you that there is no other synonym for this but Peruvian bark and 𝙬𝙎 bark. So it is also bark. Cinchona is a bark. Yes, the name itself bark. I'll give you a hint. Okay, so no other drugs are having a suffix Okay, a suffix as a bark, but these are having the Peruvian bark, cinchona bark, 𝙬𝙎 bark, cinchona bark. Okay, maybe the other drugs maybe have, but these are very familiar. Okay, coming to that biological source, it can be obtained from four sources, that is cinchona 𝘂𝙬𝙎𝙎, cinchona saxirubra, cinchona officinalis, cinchona 𝘿𝙄𝙎𝙎. But in a previous GPAT examination, they have asked like which of the source or which species accounts a major number or major amounts of the quinol alkaloids. They gave you these four options. Among these four, which one accounts or which one having the more number of quinol alkaloids? Yes, undoubtedly the cinchona 𝘿𝙄𝙎𝙎𝙎. Cinchona 𝘿𝙄𝙎𝙎𝙎. L refers for larger amounts. So you should remember like that. In order to avoid the confusion, L means larger amounts. Okay, like how you should remember this one. Okay, L means larger one. And coming to the family, Rubiaceae, family Rubiaceae. Okay, yes. So even though we are having four types of sources, but higher amounts will be obtained from L, large 𝘿𝙄𝙎𝙎𝙎. Okay, yes. Coming to the chemical constants, yes, that's what I said. In all species except cinchona 𝘿𝙄𝙎𝙎 contains highest percentage of alkaloids. That's what I said. And main alkaloids are quinine, quinidine, and they in their respective dimethoxy derivatives like cinchonidine and cinchonine. Cinchonidine and cinchonidine. So which one are the dimethoxy derivatives? They are cinchonidine and cinchonine are the derivatives. And now moving to that, they are present in the salt with phenic acid. In generally, the alkaloids are exhibits as a free basis or free salts of organic acids in plants. But what will be the major required one? We require the salt form, because the salt form has a major solubility in water. Okay, so that in generally the quinine will be present in the salt form, present as salt with quinic acid, free salts of organic acid. The salt should be present in along with the acid or an organic acid, if every alkaloid. So like how it is present as salt with quinic acid and cincotannic acid, and also cincotannic acid and glycoside like quinoline. In the glycoside also possess a minute amount of glycoside, that is quinoline. And whereas this quinidine is also obtained commercially from cuprea bark, that is Ramigia paniculata. Ramigia paniculata. Yes, such type of things to be noted and should be remembered, because so they may ask like from other than the cinchona, from which source you can get this quinidine. Yes, undoubtedly the cuprea bark. Okay, so that's about the chemical constants of the cinchona. But whereas this microscopy, I was only focusing on the main important microscopies on the every drug. So if I included any of the points in the microscopy, you should definitely remember that, because that's maybe the chance of questions that are expecting from the microscopy, especially that's what I included in the slide. Okay, so coming to that, this cinchona exhibits a different astrological properties, characteristics of bark. Why should we remember? Because we are having two barks, that is cinnamon and cinchona, whereas cinnamon is a aldehyde or volatile containing drug, but cinchona is an alkaloid drug. So, but in order to distinguish from each one in detail or each one differently, should definitely have an idea regarding the histological properties. So, for that reason, in order to avoid the similarities, morphological similarities, and microscopical confirmation, I included all these points. Yes, within the cork cell is a phylloderm of several layers, which is a polygonal shape. Which means the cork cell is a phylloderm. Within the cork cell, the phylloderm will lies within the cork cell, outermost layer. Okay, cork cambium and all these things. And cortex consists of several secretory channels, the outermost layer. Okay, and the phloem fibers. So this, the way the secretory cells will be there, secretory channel will be there in the cortex in the self. Okay, the cortex. Okay, an idioblast of calcium oxalate system is specific characteristic of cinchona bark. Yes, whenever you want to identify the microscopical characteristics or microscopically you need to confirm, should check all these things. Yes, if it is consisting of idioblastic calcium oxalate crystal, then you confirm it is cinchona, not cinnamon. Like how we need to remember. And distribution and size of phloem fiber differ in various species. Yes, with respect to the four species, the 𝘂𝙬𝙎𝙎, saxirubra, officinalis, and 𝘿𝙄𝙎𝙎𝙎, they differ in their different species. Okay. Whereas the alkaloids and starch is present in parenchymatous tissues. Okay, so mostly the starch and other alkaloids will be present in the parenchymatous tissue. So these are the diameters and thickness of the four different species. You can have a look on it. But where is the 𝙂𝙎𝙎𝙎𝙬 is this one, where it contains or it accounts for most number or larger number of quinoline alkaloids. Yes. Next, coming to that, so these are chemically these are nothing but quinidine. So these are the structures. So these are diastereomers, quinidine and quinine, and whereas the synchronidine and synchronine are the two diastereomers. So these are the structural representation right over here. You can observe the major changes of this quinidine and quinine, synchronidine, d-methoxyquinine and d-methoxyquinidine. Let us see but we have seen that cupress plant, from the cupress we can get the quinidine. Okay, but how can we get? We get from this by the addition of a phenol group. If we add the synchronidine to that, this synchronidine, if we add one phenol group at 6th position, then we can get the cuprin. Okay, so that we can commercially or we can chemically we can synthesize the cuprin. So maybe there is a chance of getting question. So from which we are going to synthesize, from which quinoline alkaloid we are going to work, from which synchronine alkaloid we are going to synthesize the cuprin, synchronine, by the addition of phenol group at 6th position. That's the thing you need to remember. And next is that uses of this quinine. We knew that it is an anti-malarial or protoplasmic poison, especially for protozoa. Okay, especially for protozoa. And this quinine is used as a cardiac depressant and used to prevent the certain arrhythmias and atrial fibrillation. And also we knew that it is a bitter tonic and stomachic. So these are the major uses. Okay, so do focus on the uses also. There may be a chance of getting questions on their uses also. But coming to that, substitutes. Cupriabark, that is a Remisia pedunculata. So this coppery red colored drugs containing quinine and other alkaloids. That's we know that already. So where we can get the quinidine from other sources is cupriabark. So that's the substitute of adulterant is cupriabark. So they may ask about sometimes they may ask about the adulterant. So which one of the following adulterant or which one of the following drug is used to adulterate or substitute the cinchona? Undoubtedly cupriabark. Cinchona cuprea, CC. Okay, cinchona and cuprea. And false cupriabark also used to substitute the original cinchona bark, that is quinine. The Remisia padiana, which contains a casconidine but not a quinine. Not at all consisting of the real components, that is quinine. Okay, yes. So coming to that, the main and most thing is a identification test for the cinchona. Quinoline alkaloids, especially the thalloquinone test is a very important test. It is a named test where it is specially meant for the identification of cinchona alkaloids. Where you can see in the thalloquinone test, if we add a powder drug to that of the powder drug, bromine water, then NH4OH, ammonium solution, ammonia hydroxide solution. Okay, here we can add ammonia or ammonia hydroxide solution. If we add, if it is produce a green color, emerald green color, that will be confirmation to the presence of cinchona alkaloids. If it will not liberates not produces the green color, emerald green color, that won't containing any of the cinchona alkaloids. Maybe it's a cupriabark, or if it is not confirms the color, that's maybe cupriabark or false cupriabark. So blood red test, which may be the dry powder of the cinchona powder. When it is added to the glacial acetic acid, it liberates the red fumes because it appears it is known as it confirms the presence of cinchona alkaloids. And now moving to that, quinine and quinidine and synchronidine. Quinine and quinidine and synchronidine, which yes, confirmation to that. So synchronidine, when it is moistened, so nothing but fluorescence test. When we subject these with a fluorescence test, especially under H2SO4, okay, under fluorescence with H2SO4, it will give us a blue fluorescence that indicates the presence of cinchona. So that's the identification test. Okay, thalloquinone, blood red test, and quinine. So this is about cinchona, and next drug is the camptothecca. Yes, so this is also one of the important quinoline alkaloid where we can see, but it has an important properties like anti-cancer drug, whereas the cinchona is quinoline drugs are nothing but there are two. One is C that start with cinchona and the other C is Campotrica. Campotrica obtained from wood of the plant, that is Campotrica acuminata. Campotrica acuminata belong to family Cornaceae or Nissaceae. Cornaceae, but it is a cancer tree. That's the reason I said that. It's the use itself saying that it's a cancer tree and happy tree. Do remember the synonyms, my dear aspirants. Yes, in the cognizance, synonyms also very important. Okay, yes, Campotrica acuminata. So this was the plant where you can see the Campotrica flowers and all these things. It's a major anti-cancer plant. Okay, and chemical constants is consisting of quinol alkaloid about 0.004 to 0.3 percentage. But when we finally extract this camptothecin, it produces and finally exhibit as a yellow needle shaped crystals. Yellow needle shaped crystals. Okay, but it does not respond to what is the confirmation of chemical test here, if at all. It doesn't confirms with the Mayer's or Dragondraft that it will be a Campotrica. Okay, yes. So coming to the uses of this plant is, we know that anti-cancer drug, and it is a prototype DNA topoisomerase inhibitor. So like how it acts on our body as an anti-cancer drug. Okay, so that's about the quinoline alkaloids. In the next upcoming session, we'll see the isoquinoline alkaloids, my dear students. Yes, until then, we'll see in the next class. Thank you. Have a great day.